mTOR signaling regulates cell growth, proliferation, and survival by coupling extracellular signals such as growth factors with intracellular nutrient availability. The mTOR kinase exists in two distinct complexes, mTORC1 and mTORC2. Intracellular nutrients and ATP activate the mTORC1 complex on late endosomes and lysosomes to stimulate protein and lipid synthesis. But, says Ivan Yudushkin from the Max F. Perutz Laboratories and the Medical University of Vienna, mTORC1 activation also relies on growth factor signaling and the mTORC2 complex.
In order to activate mTORC1, you need to remove the inhibitory signal from so-called tuberous sclerosis complex, and this is done by phosphorylation of one of the components called TSC2 by a protein kinase called AKT. mTORC2 couples the signaling from growth factors to AKT activation. Essentially, mTORC2 directly phosphorylates AKT. And the specific questions we're interested in is where inside cells the mTORC2 activity is, and how actually growth factors direct mTORC2 activity towards its substrate, AKT.
Immunofluorescence studies have suggested that mTORC2 localizes to mitochondria and the endoplasmic reticulum, but that doesn't necessarily mean that the complex is active at these locations. Yudushkin and colleagues therefore developed a reporter for endogenous mTORC2 activity based on the FRB-FKBP heterodimerization system. First author Michael Ebner explains more.
We tagged AKT, the substrate of mTOR complex 2, with FRB, and we co-expressed this with peptides that localize to different subcellular compartments that were tagged with FKBP. And upon addition of AP21967, a wrapper log, to the cells, we could recruit the FRB tagged AKT, the locator 2, as we dubbed it, to various subcellular compartments, and then do a Western and test whether it was phosphorylated or not and to which degree.
And we found that the endogenous mTORC2 activity is mostly found at the plasma membrane, at the outer mitochondrial membranes, and subpopulations of endosomal vesicles, specifically early and late endosomes. We did not find any endogenous activity of the complex at the endoplasmic reticulum where people were expecting this to happen.
The phosphorylation of AKT by mTORC2 in response to growth factor signaling is dependent on PI3 kinase. A previous study suggested that the product of this enzyme, the phospholipid PIP3, directly binds and activates the mTORC2 complex.
So we looked at regulation of mTORC2 activity at various compartments by PI3 kinase using a very specific inhibitor, and we found that actually at the plasma membrane, the activity is independent from PI3 kinase. We could inhibit PI3 kinase, and the activity is still phosphorylating our reporter. We could grow cells with or without serum. We could stimulate cells with a growth factor, with insulin or not, but the activity is exactly the same. It's not increased.
Growth factor stimulation, or PI3 kinase inhibition, did, however, affect the phosphorylation of endogenous AKT, suggesting that PIP3 production might regulate mTORC2 activity at the plasma membrane by controlling recruitment of the complex's substrate. In this scenario, phosphorylation of the locator 2 reporter wouldn't depend on PI3 kinase because it can be recruited to the plasma membrane by the FRB-FKBP interaction instead.
And in order to test whether this is true or not, we used this reversible recruitment system where we could recruit AKT to the plasma membrane, and we saw that it's actually phosphorylated exactly as we expected from our other reporter. But then we could actually add the competitor, which would outcompete the AKT from the membrane so that it becomes cytosolic. And we saw that it is actually dephosphorylated, probably due to the action of cytosolic phosphatases. So these experiments basically demonstrate that the phosphorylation of AKT by the plasma membrane localized mTORC2 is regulated by the availability of substrate and the membrane. PEP3 recruits AKT to the membrane, but the activity is already there. It's poised to phosphorylate.
In keeping with this, the lipid-binding pH domain of AKT is recruited to the plasma membrane upon growth factor stimulation, but is displaced into the cytosol when PI3 kinase is inhibited. In contrast, the mTORC2 subunit MSyn1, which also contains a pH domain, constitutively localized to the plasma membrane and didn't change its localization in response to growth factor signaling or PI3 kinase inhibition. Away from the plasma membrane, however, Ebner et al. Found that mTORC2 activity on early and late endosomes was dependent on PI3 kinase.
If you remember, the late endosomes is exactly where mTORC1 is active. This is where it senses the intracellular nutrients, and the mTORC2 appears to be trafficking from the plasma membrane all the way to the late endosomes. And this could be something which basically couples the extracellular cues with the availability of nutrients and the energy state within the cell. But again, this is just a model. We only covered the localization of the endogenous mTORC2 activity and how growth factors, by activating PI3 kinase, recruit AKT, the substrate for mTORC2, to the plasma membrane.
Yudushkin and colleagues now want to investigate the physiological roles of the different pools of mTORC2 activity, as it remains unclear exactly what the kinase complex is doing at the plasma membrane, the outer mitochondrial membrane, and on early and late endosomes. For now, though, you can learn more about the localization of mTORC2 activity inside cells in the paper by Ebner et al., published in the February 6, 2017 issue of the Journal of Cell Biology.